First of all, welcome everyone. Some of you probably saw that we had some technical difficulties. So our apologies for that. What we're going to do instead is we're going to start with the navigation and interpretation of molecular networks for GCMS. And if the stability holds up for GNPS over the next half hour, we will actually do the rest of the workshop as well. So I think that's how we're going to try to tackle it. Are there any questions at this point? Okay, great. So somewhere I had the first sort of intro slide. So this was a slide that we had to make this morning, partly because we have the technical difficulties, because otherwise we would have carried you through the entire workflow already. But basically, one of the things that GNPS does, it allows you to create molecular networks. And we'll explain in a little bit what molecular networking is if you're not familiar with it, and also how that can be applied to GCMS data. Now, we recently built basically GNPS for GCMS. And we can take any of the deconvolution outputs from any of these different deconvolution softwares, but we also have a new one called MS Hub, which is a machine learning approach for deconvolution. And if we have time to cover that, or if GNPS will allow us to cover that in this workshop, we will do so at the end, not at the beginning, because we're testing the GNPS infrastructure at this point, the function of it. But it can take any GCMS deconvolution output and create that as input for both library search as well as molecular networking within the GNPS environment. And so I think Mabel will explain how we do molecular networking within GNPS, or how that functions for GCMS. And then Alexander will do the hands-on portion of how to interpret these molecular networks within Cytoscape.
Yeah.
Mabel?
Yes, yes. Now you can see my screen. Can you see my screen?
Yes.
Okay, perfect. Okay, so good morning, everybody. So we're going to start explaining for not familiar public what actually a network is and how it's useful for different tasks that we have to accomplish when you're doing metabolomics with GC data. So usually when you're analyzing the metabolome, you can analyze different type of matrices, and usually GNPS until this moment was using only data acquired to instrumentation from LCMS. So basically what it was doing with LCMS is that you have these two dimensions when you have the retention time of the different molecular features, and you have the parent ion. And then you have the different fragmentation patterns when you are doing the MSMS fragmentation in the different instruments that you were using for LCMS. So that was the start point for doing the molecular networking using LCMS data for metabolomics. And at the end, you have different fragmentation patterns of different compounds. So what it basically was, it's happening inside the GNPS platform is that the software is aligning the different fragmentation patterns according to similarity. So what it basically does is just comparing the fragmentation patterns between the different runs and the different samples, and is developing a consensus mass spectra. And in that case, you have different fragmentation patterns when you have different fragments and also different abundances, which are included in the algorithm for doing the comparison of similarity. So at the end, you have something like this, and you have cosines because the algorithm is working with vectors. So the cosine actually is related to the similarity, to the comparison similarity between the different mass spectra or the different fragmentation patterns. And inside the network, what you basically has is a comparison of similarity between the different fragmentation patterns inside a single job. So inside the network, what is happening is that the cosines that you have here, which are basically a measure of similarity, and the different consensus mass spectra are changed inside the network. So the consensus mass spectra actually becomes a node, and the cosine value, it becomes an edge. So that's also important to highlight, is like the vocabulary that you have to be familiar inside the network. You have nodes, which are basically these circles, which represent consensus mass spectra, and the edge, which is basically a line, like is connecting different nodes inside the network. So inside the network, you have different nodes, edges, and you will have different clusters, which are basically, in this case, for example, we could say that we have two different clusters, one here with the MSD and A, and the other one with the other 5 molecular features in this case. So I'm going to show you an example of how it can be useful, a network for analyzing data. This example, it came from the Queen Laboratory, where they basically take mice and they were comparing the metabolome when you have germ-free mice and mice with specific microbes. And when you were doing the molecular networking, you can see here that the nodes that I speak before are colored between these different treatments. So the blue ones are for the treatment where you don't have microbes in the mice. The green ones are for the ones that has specific microbes, and the pink ones were the compounds that were shared between the two different treatments. So when you take a look, for example, to this specific cluster, when you have the 3 types of compounds, and we're gonna make a zoom, you can see here hit with one of the annotation of the libraries, which is this muracilcholic acid. So based on this annotation, we can take a look to the different other nodes that are close to each other, because actually they share probably some specific moieties in the structure that could be useful for doing the annotation of the other compounds here in this specific cluster. So we're going to take a look.
And if there are any questions, feel free to use the chat. I don't know if the unmute mute button works, but I think with this small group, it would be nice to have people, give the people the ability.
Okay, I'm not looking at this moment the chat, so if Ming could tell me if somebody is asking a question, I can stop and just answer immediately. So when we take a look to this specific cluster, we can find here different interesting applications of the molecular networking. So as I told you, we have one annotation, which is the pink compound, which is this acid, this bio acid, but we have other different things here, other different annotations that you can find it here with the structure. And this is very interesting because which actually is telling us combining the annotation of the compounds and at the same time the metadata, which is basically the different colors that are interesting for us and are not related directly with the mass spectrum. We call that metadata. We can see here that, for example, this compound is shared between both treatments, but there are other compounds that are specific to the treatment where the microbes are present, which is basically suggesting that probably the synthesis of these compounds could be related with the presence of the microbes. So this is really cool because in this specific study, it was the first time that they discovered this specific bile acid, which is present in mice, but it's also present in humans and could be related with some specific diseases like inflammatory bowel syndrome in humans, for example. So this is really cool because you can spot this directly inside the network. And another interesting application of the network is when you have unannotated compounds like this 517 and like this 482. Sorry. So you don't have idea because you don't have a direct match with the libraries inside GNPS, but with the network, we can at least say that probably is a bile acid, definitely, and you can start to navigate inside the fragmentation patterns and propose at least a change in the side change of the amino acid for proposing some structures for these two unannotated compounds. And you can do this more easily when you're using the network as a base information for proposing an annotation for these structures. So what happened with GCMS data? So we have similarities and also difference in comparison with the LCMS networking. So we also are comparing between fragmentation patterns, but in this case, we don't have the parent ion as we have usually for the LCMS data, because due that the fragmentation is performed with high energy. Usually we have fragmentation patterns, and in many cases we don't see the parent ion actually, that is the molecular ion of the compound. But what we do have in the GCMS data is a really good annotation possibility because the libraries are more populated, and the fragmentation patterns are also more reproducible in comparison with the LCMS instruments. So instead of the molecular ion, when you're taking a look to the network inside the GCMS data, you usually have different annotations here. And here, for example, you can see that sometimes the automatic or just the first top hit annotation should be reviewed, of course, because, for example, in this case, we intentionally put like compound 4 in 2 different nodes of the network. And that if you are familiar with GC data, that is very common when you're working, for example, with alkenes, that you are seeing, for example, the same compound in 2 different nodes or into different peaks in a chromatogram. So you have to perform like a manual revision of that annotations. But at least the new information that the network is providing us is the similarity comparison between the different nodes that are related to each other. Wait. Okay, so the other interesting thing with the GCMS data is that as the fragmentation patterns are more reproducible, you will see that actually the clustering inside the network are highly related with the structure, more related with the structure that actually is happening with the LCMS data. So we were doing this with one of the jobs that we have in the public repository. And you can see that, for example, the cluster are completely related with the chemical family when you're using classifier for for each of the compounds here. So imines are clustering completely different in a different place inside the network than to fatty acids or alkenes, for example, in this case. So that's also pretty useful, especially for these unannotated compounds, that depending of the research area that you're working on, you can have a lot of them or just a few of them. So the similarities are also visible for another not so interesting compounds, as for example, organosylicon compounds that are pretty common from the column or from the different materials that you were using for doing different extraction methods. So actually, when we're coloring this inside the network, we can take a look to all the not significant or usually not significant molecules or compounds that are extracting inside the chromatogram, but are not really informative about the metabolomics that we are usually studying. So for example, these three clusters are signalizing different siloxanes or organosilane compounds that are really common inside GCMS data. And for the other chemical entities, you will see similar patterns. So this is really informative, as I told you, when you have annotated compounds, and you can combine this, for example, this metadata, which is related with the chemical nature of the compounds, and at the same time overlay, for example, information regarding the metadata of the mice, if you have microbes or without microbes, and you will see interesting patterns as I show you with the other study. So when you take a look, for example, to this specific cluster of fatty acids, you will see like the usual procedure that you have to perform doing the manual revision of the notation when you run a job inside GNPS. So for example, when you take a look to all of this, you will see that most of them are actually fatty acids were classified according to classifier, but some of them are not. For example, this case, this case is not annotated. So at least we can say that it's a fatty acid according to the information of the network. And that was also possible without the network, but in this case, we have the support at least of the cosine value, which is really useful, at least for having like a measurement of similarity of the mass spectrum. And we have another two compounds that are not fatty acids that are signaled here with the arrows. So we can take a look closer to the mass spectrum inside GNPS and start to see what is going on with these specific compounds. And probably what we're going to figure out is that this annotation is wrong. It's a misannotation, but we have different hits for reviewing and actually saying, okay, this is not the ester that is proposing here. Probably it's a different thing. Probably it's a fatty acid, and maybe we can say something like this. It's a fatty acid, maybe with 6 carbons. So this was some of the applications that you can have for molecular network. I will take a look to the chat and try to see if you have some questions specific. Yes, the convolution, unfortunately, we cannot do it yet. I hope that we can do it later or maybe another day, but you're going to see that it's really straightforward, the steps for performing the deconvolution inside GNPS in comparison with the other platforms that are also useful, but I think that personally GNPS is more intuitive.
Thank you, Mabel. Let me share my screen so we can continue with the hands-on portion, how to do these networks. So any questions at this point, because there were quite a few questions in the chat. So if you guys have any additional questions regarding that, please type it up in the chat, and then we're probably gonna have more questions going forward when we do the hands-on portion. So let me share my screen. So as Peter mentioned, the original plan was to introduce the deconvolution for GCMS data, and then molecular networking and library search job. That is a separate workflow. And the result of that workflow is a set of annotations for the features that are found in the samples. And then we can download a Cytoscape file and do molecular networking afterwards. So basically we have to fast forward straight through to the last part, the building molecular networks using Cytoscape. And the network job that has been completed is given in the Google document that I believe everybody has access to. So just in case anybody doesn't, please send a message through the chat so we can share the link. And for everybody who is at the document, please go ahead and click on this link right here, the GNPS link for the library source of the full data set. And it will take you to the GNPS page where the job has been completed, which is this page right here. So once you get to that page, click on download Cytoscape data link right there.
Also, if you have a hard time following, just let us know and we will slow down.
Yes, please do. I'll try to go slowly, but in case there's an issue or you're not following, let us know. So when you do that, the download should start automatically. And if it doesn't, click on download link. You should be able to download the zip folder right here. So once you do that, go ahead and unzip the folder. You need this file inside this folder, the GraphML file that contains the network that has been generated by the library source workflow. So this is the file, so I'm going to take it and I'm going to put it in folder where I can work with it. That will be my file. Okay, so now to visualize the network, we need to use Cytoscape software that is a third-party software that you should have been downloaded prior. So the Cytoscape that I have installed on my computer is the little bit older version, 3.7.1. There was a question in the chat whether the Cytoscape installation throws up the error about Java runtime environment, and the way I was able to solve this problem is to roll back to the earlier version, 3.7.1, because it doesn't have a couple additional functionalities, but we're not going to use them today. So the earlier version will work as well if you're struggling with the Java runtime environment error. So go ahead and launch the Cytoscape software. And when you do, you should see this window right here. Okay, so all good. So once you opened up the Cytoscape, we're going to load the file that contains the network. The file that contains the network, which is downloaded, can be imported by going to File, Import, Network from File. And this is the same procedure that we would do for LCMS data for molecular networks, such as feature-based molecular networks that we had a workshop in weeks prior. So basically, you do the same sequence of steps. You click on Network from File. And then you select the network file that we just downloaded and unzipped. So this is the file that we just downloaded right here, the GraphML file. So click Open. And the Cytoscape will start importing the network from this file. Okay, so here's the network. It's loaded up, and as you can see, it appears similar to LCMS network. There are some clusters with a high connectivity. There are some clusters that are fairly small, and in this case, we do not import singletons because they do not since they do not connect to each other. They're not very informative. But the network cluster topology is somewhat similar to LCMS. You can see that there are some like very expansive clusters that are highly interconnected, so that those must be chemically related compounds. But the appearance of the network right now is not very informative, so we're going to go through a sequence of steps to make it look better. So the first thing we're going to do is change style to something that's more palatable, because the default style of Cityscape is doesn't look very good. So you go to Style tab, and then you click on the Style drop-down menu right here. It will pop up this selection of different network visualization options. And this is really up to you which one works best for you, which one is most visually appealing or more informative for your data. And you can play with all kinds of different styles and see what the resulting network look like. Let's say in this example, we're going to use the sample 1 style. So select that, point mouse to it, and then click on it. So the network appearance has changed according to that style. So what we're seeing right now are our compounds labeled by the feature ID. And feature ID, this is just a number that has been assigned by the deconvolution software when the spectra were processed from the raw data. So there is this notch on the edge because we don't have any information displayed for edges. And in this case, probably it's going to create sort of messy appearance. So maybe it's best to not display anything on edges. So to do that, we can go to Edge, Label, and then click on the little garbage can icon, which will delete the label. So remove. So now this little notch went away. Okay. So far so good. Any questions? Okay. So the next thing we're going to do is we're going to go to Node, and we're going to change the number to something more informative. And this is the main difference of GCMS data from LCMS data that Mabel has mentioned, because for LC data we have really low annotation percentage, maybe low single digits. We most of the time not going to have annotation for the node. But in GCMS situation is quite different. We have annotation in essentially 100% of cases, because libraries are really large, and there's a really high chance that at least something in the library will match to our spectral pattern. Therefore, there will be at least some annotation available for every node. But the question is whether this annotation is correct. And this is something that network can help us with. So to display annotation on your node, you need to go to Label, expand it, and click on the column right here. And then we need to select the column that's called compound underscore name. And it has to be pass-through mapping, so that's already selected as a pass-through mapping. So now you see that we have compounds labeled by their name. And when we do that, we immediately see that the patterns are observed in the data, similar to what Mabel has shown you for the reference spectra from this library. We see, for example, here hydrocarbons are clustering all together. So all of the hydrocarbons are assembled by the network in this really neat cluster. So unfortunately, because we didn't go over the data, we don't have a sort of like a background, a rationale, what we're trying to achieve in this particular network. But just to give a really quick summary, these data are breath analysis experiments for the esophageal and gastric cancers. And in this study, the researchers who collected this data set, they were trying to analyze which molecules are potential biomarkers of esophageal and gastric cancers. And the categories they were particularly interested at are gastric cancer and healthy controls. So one thing we can do is we can color these nodes according to the category they belong to. So in this case, it will be cancer and control. So to do that, we need to go to the image chart and click on the left icon, double click. So the window gonna pop up that looks like this. And we're gonna do a chart. So click on the chart tab. And the categories that we're looking at are cancer, OGC, which is esophageal gastric cancer. So select this category and click Add button. This category will be added. The other category is control, so the healthy patients without cancer. So select that category and add selected. So the charge that is there by default is not needed, so remove that. Okay. So one other thing we can do, we can customize colors that are going to be displayed. So if you go to this tab right here, it's called options. We can select colors. So the first color will be corresponding to the first group, which is OGC. The second color will be corresponding to the control group. So let's say cancer, we can keep red, but for this one, let's say we're going to select green, other color. So now the OGC will be corresponding to the red and control will be corresponding to green. One last thing that we need to do is we need to change the chart type. So there are multiple chart types right here. The one that we're going to use here is the pie chart. So make sure to select the pie chart tab. So once you do that, click apply. And now the network is colored by the abundances of this particular compound in that category of the metadata. So when it's OGC, it's going to be red. When it's control, it's going to be green. So we can immediately see that some compounds are higher in OGC than in controls. One other thing that we can do is to display information such as, for example, molecular abundance. And we can do that as shape or size. Size is probably most informative, so let's do that. So to change the size of the node, we need to go to the size right here, expand it. Don't confuse with the label font size. The font size refers to the size of the text, but we're going to be changing the size of the node. Okay, so once you do that, in the column, click on the select value to get a drop-down menu. And we're going to use abundance of the ion of this particular feature. And the abundance of the feature for GC would be sum precursor intensity, which is the sum of all the peaks in the spectrum, and that's a proxy for the total abundance of the of that particular feature. So select this category. Now we need to select the mapping type. So the mapping type for this had to be continuous mapping, which is the first option. So select continuous mapping, click on it, and it should give you this this plot right here. So if you double click on the plot, it will make it large, so we can actually play with it and change the values. So this is the plot. So what the plot shows is the smallest node that we have, which is the least abundant compound, would have a size of 10, and then the largest node, which is the most abundant compound, would gonna have abundance of 30. it's still pretty small. So let's make it a little bit larger. So the minimal size, let's make it 20, something like this. That's gonna look much better. And then for the large node, we can make it much larger, so it would be very conspicuous that something is very abundant. So let's make it as high as, let's say, 200. We can also have nonlinear dependence. So to do that, we can do this. You can click the Add button, and we'll add another notch. So this box can be slid up and down, and this triangle can be slid up and right, left and right. So let's say we change it like so. So now our most abundant compound is going to all look very large. Like this. Okay, once you've done that, click OK. And now the network node's going to get resized according to the values that you're given to the network. So now if we explore the network, it's going to look a little bit different. So most of the nodes are still small because those are minor compounds and they're present in really low abundance. Many of them might be just the impurities or even deconvolution artifacts. But some compounds are going to look very large. Like here's an example of compound that's really, really abundant. And that gives you a really easy way to visualize which compounds are abundant in your sample. Like this one, for example, that's obviously because this is trimethylsilyl, that's probably the column bleed, which is very common in GC when you have your background contaminants such as siloxanes are being very abundant. Here's an example of another abundant compound, and that yet another siloxane. But for example, here, there's a very abundant compound. I want to say a couple words about this one. This is the most abundant compound in the data. The issue with this, so this compound is actually an internal standard that has been spiked by the researchers when they were doing their the sample collection. This annotation is incorrect. This actually D8 Toluene, but because we only use public libraries in our search, some of the compounds that are more, I should say, want to say exotic, the ones that are, let's say, maybe synthetic, they are not very well represented. So there might be misannotation for those compounds. Most of the biological compounds that are, more common, they're going to be annotated correctly. And then the size of the library. So if you add your own personal library, and you do search against public libraries and your library that maybe, for example, commercial in the library that you collected yourself, in your laboratory, the size of your library will dictate whether this annotation, basically correctness of your annotation. The more expensive is your library, the higher is the chance that every single compound is going to be annotated correctly. But at this point, we see that with the public libraries alone, most of the biogenic compounds are already annotated correctly. Okay, so one thing that is also possible to look at these networks is if you go to View and Show Graphic Detail.
So there's one question, that's come up for a couple of people that are interested in this in terms of Classifier. How do you use Classifier onto these networks?
Okay, so to use Classifier on these networks, we use the MoleNet enhancer algorithm developed by Madeleine Erst and Justin Vanderhoft. It is implemented as a option on the workflow for CMS data, but for GCMS data, it has to be used as a separate script at this point. Eventually, we'll probably get incorporated as well. So to get there, we have a link on the documentation for GCMS. If you go to to GNPS tutorial and you look for GCMS data, you will have a link in there for the MolNet enhancer and script to run. And we should probably paste it in the chat as well, so it will be easier to access.
Okay, I will look for them and paste it in the chat for everybody.
Yeah. Okay, so if you select the view graphic details, when you zoom out on your network, it preserves all the graphic details, and you will be able to see the network without nodes getting boxy. So like, for example, if you want to use network as an image in your paper, that is the option to use to have a good looking network. Okay, so far it has been basically just visualization steps. We haven't done any real chemistry yet. So I just want to show you really quick couple examples how these networks can be used for your chemical analysis. And one thing I want to point out, if you go to this workshop document, at the bottom of the document, we have several videos, including one done by Mabel, where you can see how these networks can be used for all kinds of additional analysis, such as visualization of your distribution according to metadata, how you can use that for discovering new compounds, how you can use that for spotting correct notation. So I do encourage you to look at those videos so you can have a better idea how you can use molecular networks. And I want to emphasize that because this is a fairly new tool and it's only been developed recently, we're still finding out what are the best ways to use that, and the ways that are suggested here by no means are exhaustive, and I'm certain that once you start looking at your own research applications, you might come up with the clever new ways of utilizing these networks. But I'm going to show you just a very basic way to use these networks for GCMS data. So one of the things that I want to show you is to identify distribution according to metadata. So again, in this case, we have cancer and control samples from patients, and we want to see which compounds are differentially expressed in cancer patients versus the control patients. One of the compound classes that is known to be differentially expressed is aldehydes. So if we're going to locate the cluster of aldehydes and look at the distribution, they might inform us on what the distribution of aldehydes for OGC cancer is. And there are several ways to navigate the network. You can just scroll it through and look at the different notations. And then you can also search for a particular compound in here, in the bar, in search bar in the upper right corner. So if you type name of the compound here, it will search for you. Or the other way to find compounds is by selecting it from the drop from this menu right here. So here it gives you information about every node that there is a network, and that includes its name. So the shared name, that's just a feature number from the GNPS job. It gives you the annotation. It gives you smiles if it's available, and abundances and everything. And there is a link to GNPS job as well, so you can go there. Okay, so in this case, we can navigate to aldehyde cluster right here, and you can see that all the hydes are really nicely clustering with each other. And again, red corresponds to the OGC cancer, and green corresponds to the control healthy patients. And from this distribution, it's immediately clear that aldehydes are indeed upregulated in cancer. So there's a slightly higher distribution for aldehydes, and it's very consistent for all the aldehydes that are present in this cluster, which indicates that it's not 100%, but it's indicative of that possibly all of these aldehydes are formed in the same way because of similarity in distributions. And it's known that in OGC in the cancer, in the gastric cancer and actually other types of cancers, there is a really high oxidative stress, which leads to lipid peroxidation, which results in production of long-chain alcohols, aldehydes, and hydrocarbons. And so in this case, our data do corroborate that that is indeed the case, and we do have a upregulation of aldehydes. And that work offer very easy and convenient way to visualize that and explore this type of information. Okay. So any questions at this point? Okay, so the other example that I want to show is to help spot incorrect annotation using molecular networks, or rather than spot incorrect annotations, is to help us to rearrange annotations that we get for each node to figure out which one is the most likely. And the way we can do that is by using network to our advantage, because molecular networks cluster compounds based on similarity of their spectral patterns. So all the aldehydes have similar spectral patterns, and because of that, they form this really nice neat cluster. So if a compound is aldehyde and it's clustered with aldehydes, but it gets incorrect annotation, this is something we can spot because this annotation will stand out. So the example of that would be this compound right here as an example. So if you look at annotation of this compound, this is an ester that is clustering with aldehydes. So that is suspicious. And to investigate whether this annotation is correct or not, we can go to GNPS job. So if you go to the link that you downloaded the molecular network from. So this is the job, the one that is given in the Google Doc. So right here. So click on this link again to get to this page of the job. And in the original workshop, we're planning to explore how to navigate this interface. So because we're going to do it next time, I'm just going to very quickly show how to navigate to this particular compound that we're interested in. So click on View All Library Hits. Once you do that, it's gonna, so because the results are pretty large, it's gonna give you this screen. So click on Show Results. So it's going to take you to the result page from the molecular network library search job. And you can see there's a lot of features in there that some of them are actually just noise that we can easily filter out, but this is something I'm going to show you how to do another time. So we want to look closely at this feature right here that we suspect is incorrect because it clusters with aldehydes, but it's not aldehyde. So if you click on the feature itself, below, you'll see the information for this feature. So this is the shared name that corresponds to the feature number that has been given to it by the deconvolution algorithm. And this is the reference number that we can use to find this feature in the GNPS job. So 956 is the feature we're interested in. So we go to the GNPS job page, and let's select this feature. So type 956, and click Filter or hit Enter. So the GNPS going to give us all of the possible annotations for this feature that we have. And on the step, at the step where we're launching this job, we selected top 10 annotations for every feature. So basically all the possibilities within top 10 are given for each feature, and then we can browse through them and identify which ones we believe, which ones we don't. So as you can see, by default, features are sorted by the cosine value. So this is the match score. And the cosine value is the highest for this annotation, O ester. However, the next one also has a really high cosine, but because it didn't float to the top, the node in the network got this annotation and not this one. And because we believe that this one is incorrect, and it might be aldehyde, we want to take a closer look at this feature and see at this annotation and see if that's the actual correct one. So to manually examine how your match looks like, you can click on this icon, and it's going to give you a mirror plot of the query spectrum, which is top, and then the library spectrum, which is the bottom. So it looks pretty good, but not perfect. If you click at the bottom one, the second one, the aldehyde, then we can say that this is also a good match, and perhaps it was better than the other one. Obviously, we have some mismatches at the higher mass range. That is indicative to me that it's probably an aldehyde, but it might be a different aldehyde, not hexadecimal, but some kind of aldehyde. And this is the utility that you can derive from the GC network. So basically, if you look at your network, you see that this is an aldehyde cluster, and this is likely aldehyde. We're not going to be 100% certain that the correct annotation of aldehyde is hexadecimal or something else, but we can be pretty certain that this is some kind of aldehyde. And so if we are investigating this holistically, not biomarker based, but what compounds in general are upregulated for OGC, we can say that this compound is clearly upregulated for OGC, and this compound is some kind of aldehyde. So we can make a statement that aldehydes are indeed upregulated for OGC. And then we can further investigate what this compound is by, for example, expanding our search libraries or including Kawat's retention index into consideration, and that's going to help us to narrow down list of potential candidates. And this is something that you can do individually for specific compounds you're interested in, or you can do it on larger scale for groups of compounds, and you can color your network by the chemical class, and you can see how chemical classes are different. Because, and this is the thing about GCMS that I want to emphasize, we are pretty certain in many cases which chemical class this compound corresponds to.
It's a question, what's the cosine cutoff for defining the nodes, or presumably for defining whether two nodes are connected?
Oh yeah, okay, so in this case, the cosine is 0.7, and this is something that a data specific. I would say the good guideline is for non-derived samples to use about 0.7 or so, and for derived sample to drop it down to maybe 0.6, because for derived data, the spectra are a little bit more variable, and similarity are not as great anymore. But it's data specific.
We need to be a bit more specific on this. So for LCMS, we actually have an FDR estimator, and this is something we published with Sebastian Booker. It's called Passthrough. It's in Nature Methods that we publish, and we can actually estimate sort of false discoveries based on spectral library matches. However, we can't do that here. And so in my experience with looking at this GCMS data, it certainly is going to be data set dependent. And so what you have to do is basically screen it, test the different cosines. But the nice thing is you don't have to rerun the network. You can just display the network based on the cosines. But so if you start to get a lot of things that don't make sense, so if you all of a sudden get these alcohols and they start to match to steroids and lots of other funny things, then you know you were too liberal with it. If you start to get really nice, clean networks where all of the networks itself make sense in terms of the families of molecules that you see, so in this case we see lots of aldehydes, then you have more confidence in it. In my experience, what I've seen so far is that when you have do silation, the behavior of GCMS and subsequent deconvolution is always poorer. So you're not going to get as great of a result compared to when it's underivatized. My opinion is, if you're worried about it, increase it to cosine of 0.85. That's what I would personally use if you want to be very rigorous about it. But then you can carefully inspect the data if you can tone it down and move to 0.7. But I would not go below 0.65. And maybe Alexander, can you pull up the slides that you had for module 1 real quick?
Yes.
Because it shows some really nice graphs to show the impact of cosine with respect to false discovery rates. And so this was determined based on known compounds that were added, present in the sample.
So, give me a second. Yeah, thank you, Peter. That's a very good point. And we actually did some testing on the optimal cosines, but as Peter said, this is data dependent, and we can give some starting point. But ultimately, it would be up to you to determine for your specific data what are the best values to use. This is the slide, I believe.
Yeah, if you can make it full screen real quick.
Yes. So this is something that we're planning on covering in this workshop, but because of the technical difficulties, we'll do it next time. But this is a graphical representation of the accuracy of our annotation, the FDR, full discovery rate percentage. So the lower is FDR, the better, the higher confidence we have in annotation, as a function of cosine score and this new metric called balance score that we introduced for the new deconvolution algorithm that we're using on GNPS. And we'll talk about it next time. So ignore balance score for now and only look at the cosine score. So basically, if we are focusing, this is...
I want to know what balance score is. It's basically one of the things that we've done is there's a new deconvolution method that we have. It's basically allows you to search for features across all files, and then how well that is explained, that particular feature, is essentially a quality metric, and that's the balance score. But what's nice about the approach that we have is that we don't need any input parameters. So you don't need to know trailing, for example. You don't have to specify that. You don't have to put the width of the feature associated with it. No, that's automatically deconvoluted in our approach. But with the balance score, we can set it to a certain percentage of quality of data that you're willing to include in your analysis. And then we can specify the cosine score. And so Alexander, you can take it from there.
Yeah, so the balance score is essentially confidence in our spectral pattern. So normally for GCMS data analysis for matching to spectral, you only use cosine score or other metric for the spectral matching. So the higher the spectral matching score, the more you're confident in your annotation. The problem with that is if you do have a spectrum in the library that's similar to your spectrum and is going to give a higher match, it doesn't mean that that's correct, because you might have a poor spectrum that just by pure chance happen to be similar to something. The balance score gives you orthogonal metric, confidence in the actual spectral pattern. So when it's high, you're certain that this is a high quality fragmentation pattern that is consistent from experiment to experiment, from sample to sample. And so that can be used jointly with the match score to filter your annotations, and we'll show you how to do that next time. But to the earlier question, what is the cosine score to use as a threshold? So if you only use cosine score by itself, like beyond 0.85 whereabouts, you're getting into really low FDR. So basically, that's where you want to be. So I guess that's the threshold. But again, it's data dependent. So in this data set, that happened to be 0.85, but in your specific data set, it might differ. If you use it in conjunctional balance score, then you can almost certainly ensure that you're gonna get only the lowest FDR matches by keeping balance score really high as well, really restrictive, about 80%. But again, that's a question for another day. So I hope that answers the question for the cosine match.
You said great, thanks.
All right, excellent. So so we're past an hour, so that would be a good stopping point, and we usually have a Q&A section afterwards. So if you guys want to stay behind and ask questions, you're welcome. If you guys have any additional questions and you want to learn more about...
There's a question about the NIST library. So it shows that the search library has static libs NIST E14. So we are not allowed to provide release NIST data to the public. But what we can do is we can provide matches to NISTs and annotate based on those, and we can make those libraries public.
Yeah.
So because they don't own those spectra. Wiley is the same thing. We can do the same thing with it. And then we have downloaded all of MassBank North America, Fiend Library, and all those other libraries. And then also a part of the release of the GCMS, GNPS infrastructure, we have an additional 20,000 reference spectra that are freely available. And so the total number, do you remember, Alexander, how many we have?
It's over 50,000, give or take.
Can you show the overlapping comparisons to NIST 14 and 17 and Wiley?
I don't have it on slides, so not easily, no.
No, just pull it up in the response document to the paper.
Okay, I need to look for that. But before I do that, I want to make a quick point as well. So if you look at the document, the Google Doc, we have a link for submitting reference spectra. And this is something that I wanted to bring up as a point for the discussion for the library search, but that would be an appropriate place to bring it up now. So basically, if you have your own personal libraries, they can be converted to GNPS compatible format, which means the spectra should be as MGF file, and then descriptors should be as a CSV file that accompanies it. And this spectra can be submitted as a batch to GNPS, and you can use it for personal tasks, or you can share with the community. So you can make it public, and then everybody will have access to the spectra. And this is something that we really do encourage people to do. And there were quite a few libraries going around, like at the institutions, for example, that could be put together as a library of sufficient size that can be shared with the community. If you are interested in using your own commercial libraries, NIST and Wiley, they can also be converted to the same format and uploaded to GNPS. And you can use it in your own private space because those are protected by copyright. They should not be shared with anybody else, but you can use them for your own personal purposes. That's what we've done with our NIST and Wiley libraries. We converted them, uploaded them, and then people who have access to them can add them to their searches. If you don't have these capabilities, you can still use public libraries that already going to give you a really good annotation rate. But like I showed in an example of the DA Tallinn, they might miss some of the compounds. Okay, so the annotations.
I found it. So we have about 70,000 reference spectra, GCMS reference spectra in GNPS at the moment. So this includes MONA, Fiend Library, HMDB Library, and then the 20,000 new reference spectra that have been released. And so with Alexander, or I can even share this.
Yeah. I disconnected.
So if you can unshare real quick.
Yep.
Sort of to show what impact that has. So if we compare the unique matches associated with public libraries available on GNPS. This includes HMDB, MONA, and all the other ones, compared to commercial libraries. And there's two different data sets that we looked at. And the first one is a data set where there were spiked standards in it. And whereas the second one was a data set, oh no, never mind. These are two data sets that are in the public domain. One is a cancer detection from breath analysis. The other one is a Lycopir aging reference. And these are just the number of annotations that were ranked, the correct annotations that were ranked top 1. So it shows a different picture in terms of top 10, but it's the first rank being correct. And what you can see is that the public libraries that we now have, this is the number, sort of 59 are only found in public libraries. There are 64 that are overlapping, and then 95 are available in the commercial library. So it clearly shows the benefit of using both commercial libraries and the public libraries. But it also shows that the public libraries have additional unique molecules that are not present in the commercial libraries. And so it really leverages both of those. My expectation now, there's starting to be a good infrastructure where we can start to collect reference spectra, that this number is growing very quickly, going to be growing quickly over the next few years as well. Because one thing that GNPS does, it leverages data that people are observing. And so if there are a lot of compounds in commercial libraries that are never observed, and so and within GNPS, we're going to grow a lot more with compounds that are observed. Is that clear to everybody?
So there was this question in the chat for the spectra addition. How is it different for GCMS versus LCMS? It's actually the same exact thing. The only thing that's different is for the data type you select GCMS, but structure otherwise is exactly the same. So Mabel has shared a link to workshop how to do spectra submission. So you can follow exactly the same sequence of steps for GCMS libraries.
Yeah, and it's also listed in the link when you go under GCMS data or GCMS analysis. So one question was, are most libraries entries based on derivatized compounds, for example, phen or MONA, or not?
So Mabel has answered that, but basically we have both. The phen libraries is mostly derivatized. MONA is a lot of them derivatized. The other ones are not, and the Borisov library, that's mostly common compounds, you know, such as hydrocarbons, you know, aldehydes and things like that. They're non-derivatized. So we got both. Okay. So the very last point I want to make is in the Google document, we do have a link to both tutorial for using workflows, and we have a link to a tutorial on GNPS website. So before the next workshop where we're going to show how to use deconvolution and library search workflows, if you want to look it up and you know, follow tutorial how to use these workflows, you might be able to do it yourself to get to this point of molecular networks, something that showed today. But hopefully we'll be able to show it to you in greater detail how to use those workflows soon. So at this point, if there are questions for people who want to stay behind,